Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad MSc Entrance Examination for the subject Biochemistry conducted in the year 2021, and I will be discussing only the biology part. Some maths and physics questions have also been asked which I have not attempted. So if you are new to my channel, please do like, share and subscribe. So coming to the first question. The key regulatory enzyme in glycolysis is, it is phosphofructokinase. Answer is option C. Then glutathione is a tripeptide containing which of the following amino acid? So it is glutamic acid, cysteine and glycine. Answer is option A. Thiourea contains. Now formula of thiourea is NH2 C double bond S NH2. So it contains two NH2 groups and a C double bond S. So answer is option B. Which of the following option have both enzyme required for gluconeogenesis? Sorry. So if you see option D, it has fructose 1,6-bisphosphatase and pyruvate carboxylase. So answer is option D. Then phospholipid bilayers are permeable to. They are permeable to oxygen, water and glycerol. Answer is option A. All transaminases, they use pyridoxal phosphate. Answer is option B. A single water molecule forms dash hydrogen bonds per bond at a time. So it forms 4. Answer is option D. Given below are different kind of reaction. From the options given below, select the option that represent the reaction that cysteines undergo. So cysteines, they undergo oxidation reaction and disulfide bond formation. Condensation and synthesis reaction, no. So answer is 1 and 3 only. So the answer is option B. The next one. Given below are statements on the torsion angle of a chemical bond in a molecule. So angle from the options given below, select the option that represent the correct statements. So angles between two planes, that is correct. Then describing the geometric relationship between atoms joined by chemical bonds, that is also correct. And angle between three atoms, no. Angle between two planes. Then planarity of the peptide bond. So answer is 4. So 1, 2 and 4 are correct. So option B is the answer. Then the complement system does not directly participate in which of the following defense functions of the body? Select the most suitable answer. So it does not take place in antibody generation. Does not participate in antibody generation. So 11D. Then which of the following is a mismatch in PCR reaction? Primer oligonucleotide, synthesis 5' 3' direction, substrate NTPs and polymerase is tag polymerase. Substrate NTPs is the mismatch. It is a DNA template. So answer is option C. NTPs are used by tag polymerase to synthesize new DNA. Next one. Okay. The signal sequence on proteins to be transported to the lysosome has. It has mannose-6-phosphate. This is a repeated question not only for biochemistry but for integrated MSc PhD as well as for other MSc entrance examination of this particular university. I have seen this question being asked. So it is mannose-6-phosphate. Which of the following does not have cell wall? So it is plasmodium that does not have a cell wall. Then phenomenon that some cells evoke a specific developmental response in other cells is called. It is called as embryonic induction. Answer is B. In behavioral science, the term taxis is used to refer to movement of an organism to an external stimulus. What is the kind of taxis that can be exemplified by birds that navigate using memorized landmark? Select the most suitable answer. So it is called as mnemotaxis that describe migration through the use of landmarks. Answer is option B. Then NADP plus is reduced to NADPH in. So in pigment system 1 and non-cyclic photophosphorylation, NADP plus is reduced to NADPH. Then Rh negative mother had an anti-Rh response following the birth of her Rh positive child. During her second pregnancy with Rh positive child, it was observed that the maternal anti-Rh antibodies coated the fetus red blood cells. What is the isotype of these antibodies? So erythroblastosis fetalis is caused by destruction of red blood cells of the neonate or fetus by maternal IgG antibodies. So here it is IgG gamma. Answer is option B. Then given are some nucleotides. Select the option which has a nucleotide that causes feedback inhibition of PRPP glutamyl aminotransferase. So it is 2, 3 and 4. AMP, GMP and IMP. So the answer is option D. This is a direct question. Then the basic unit of classification in taxonomy is species. Answer is option C. Coelentrates with respect to body cavity are they are acoelomate. Answer is option D. Which of the following will not affect the Km of an enzyme? So it is increasing the enzyme concentration. That will affect only the Vmax. Vmax is dependent on enzyme concentration, not Km. Then for monoclonal antibody production, antigen specific plasma cells are fused to myeloma cells followed by selection of fused hybridoma cells. Which of the following is a critical step specific to this process? Select the most suitable answer. Now plasma cells plus myeloma cells on fusion, you will get hybridoma cells. These myeloma cells, they lack HGPRT, an enzyme that allows the cell to grow in the HAT medium. Now upon fusion of these hybridoma cell, this plasma cell and myeloma cell, you will get three type of cells. That is unfused myeloma cells. They are HGPRT deficient. Unfused plasma cell, they have short lifespan, and fused hybridoma cells. Now how will you select for these? Then in mammalian cells, DNA synthesis is carried out by two distinct pathways, that is de novo synthesis pathway and the salvage pathway. And de novo synthesis pathway, it depends on the activity of dihydrofolate reductase. And salvage pathway, it depends on the presence of exogenous hypoxanthine and thymidine and the enzyme HGPRT and thymidine kinase. So you are using the HAT medium for selection of these particular cell. Now HAT medium, it is a amphoterine dihydrofolate reductase inhibitor, and it inhibits de novo nucleotide synthesis pathway. That is by it will inhibit the activity of dihydrofolate reductase. So while exogenous hypoxanthine and thymidine are supplied to ensure DNA synthesis can be carried out via the salvage pathway. So if you supply with exogenous hypoxanthine and thymidine, then even though the de novo pathway is not working or not is inhibited, you can carry out the synthesis of DNA via the salvage pathway. Then unfused plasma cells are easily eliminated as they do not replicate in the culture medium. Then unfused myelomas. So myelomas, I told you, they lack HGPRT. So in the case of unfused myelomas, aminopterin blocks the pathway that allows for nucleotide synthesis. That is, it blocks the de novo synthetic pathway. Hence unfused myeloma cells, they die as they cannot produce nucleotides by the de novo or salvage pathway as they lack HGPRT. Then fused B cell myeloma cells, they survive since the HGPRT gene coming from the B cell is functional. These cells produce antibodies and are immortal. So now we have to select the suitable answer. Frequent changes of cell culture media to avoid contamination. No. Then use only those myeloma cell which has hypoxanthine guanine phosphoribosyl transferase, that is HGPRT, and functional pathway of salvage synthesis of nucleic acids, because myeloma cells they lack HGPRT. Then use of thymidine in the cell culture medium. We have to use thymidine, exogenous hypoxanthine and thymidine. Then treatment of cells with aminopterin to disrupt de novo purine synthesis in myeloma cells. Yes, that is correct. So answer is option D, because fused myeloma cell hybrid survives since the HGPRT gene coming from the B cell is functional. These cells produce antibodies and are immortal. So the answer is option D. Then given below are molecules and the nature of inhibition to their respective targets. Select the option that is incorrect. So cyanide act as a non-competitive inhibitor to the enzyme cytochrome C. Actually, cyanide is a competitive inhibitor, so that is the incorrect statement. So answer is option A. Then from the options given below, select the set of molecules for which Hill coefficient will be less than 1 for oxygen binding to hemoglobin. So it is 2,3-diphosphoglycerate and hydrogen ions. Answer is option. Then next one. Which of the following is not present in normal urine of human? So normal urine of human, they contain urea, creatinine, water, some inorganic salts, then some organic compounds, and then some salts also. But proteins and bacteria are not present in the normal urine of a human. So both A and C. Answer is option D. A researcher wanted to study initiation of protein synthesis in bacteria by using specific inhibitor for initiation step of translation. Which of the following should the researcher use to treat the bacterial culture for the study? So answer is tetracycline, because they inhibit the initiation of translation in a variety of ways by binding to the 30S ribosomal subunit, which is made up of 16S rRNA and 21 proteins. Then chloramphenicol, it prevents protein chain elongation by inhibiting the peptide transferase activity of the bacterial ribosome. Then puromycin, it results in premature termination of translation, and cycloheximide interfere with the translocation step and thus blocking translational elongation. So here he has to use tetracycline for his studies. Then considering the interspecies interaction as positive if it is beneficial and negative, if it is harmful and null, 0, if neither harms nor helps. Which of the following relations are incorrectly presented here? Select the most appropriate option. So the option is parasitism plus minus, commensalism plus plus, mutualism plus plus, predation plus minus. The answer here is commensalism. It is not plus plus, it is plus. It is 0 and plus, because it is a relationship between individuals of two species in which one species obtain food or other benefits from the other without either harming or benefiting the latter. So it is 0 plus. Then next one. When data distribution is normal and is skewed to the left, it indicates that the mean is less than the median. Answer is option D. Then plasma volume of 8 adult males are as follows: 2.75 liters, 2.86 liters, 3.37 liters, 2.76 liters, 2.62 liters, 3.49 liters, 3.05 liters, and 3.12 liters. What is the median? So first arrange the data in either increasing or decreasing order. Now median is defined to be the mean of the two middle values, because these are even numbers: 1, 2, 3, 4, 5, 6, 7, 8. So 8 values are given, and that is 8 is an even number. So median is defined to be the mean of the two middle values. So 2.86 plus 3.05 divided by 2, you will get 2.96 liters. So the answer is option A. Then which of the following is an allosteric effector of myoglobin? It is lactate. Suppose FADH2 is the electron donor to the electron transport chain of mitochondria. How many protons will be pumped into the intermembrane space from the matrix of the mitochondria when one oxygen molecule is completely reduced? So one FADH2 pumps 6 protons are moved during one turn of the citric acid cycle. But each glucose produces 2 pyruvate molecules which are converted to 2 acetyl-CoA molecules. And each acetyl-CoA molecule results in one full turn of the citric acid cycle. So 2 acetyl-CoA results in 2 full turns of the citric acid cycle. So 2 FADH2 and 12 protons are moved. Answer is option C. 12 protons. Then cardiolipin is enriched in. So it is enriched in mitochondria. Answer is option. Then compared to absorption of light, fluorescence. So it occurs at longer wavelength and with a lower energy gap between final states. Answer is option A. Then chromosomes are best observed in which phase of the cell cycle? So it is the metaphase stage. They are more condensed there. Answer is option D. Then thymidylate catalyzes the conversion of DUMP to TMP using which of the coenzyme? It is N5-10-methylene THF. Answer is option C. Maple syrup disease is due to the defects in metabolism of which one of the following amino acid? It is valine. Answer is option A. Then given below are some statements on intracellular receptors of hormones. Intracellular receptors of hormones are present in the cytoplasm only. Intracellular receptors of hormones are present in cytoplasm and nucleus. That is correct. Intracellular receptors of hormones possess transcription factor activity. Yes. And intracellular receptors of hormone are stabilized by heat shock protein, of course. So 2, 3, 4 are the only correct options. So answer is option D. During cryopreservation of cells, cryoprotectants, they lower freezing temperatures and increase viscosity. Answer is option D. Next one. Which pituitary hormone regulates glucose homeostasis by antagonizing the insulin action? It is growth hormone. Answer is option B. Excess secretion of human placental lactogen during pregnancy causes. It causes gestational diabetes. When neurospora of opposite mating types auxotrophic for leucine biosynthesis were mated, the diploid was prototrophic for leucine. This is a case of It is a case of complementation of mutant allele. Answer is option C. Then, the F1 offspring of Mendel always looked like one of the parents because one allele was completely dominant over the other. Answer is option A. Then, Huntington's disease is caused by a dominant mutation. If one of the parent has the disease, what is the probability of the offsprings will also have the disease? So Huntington's disease, it is caused by a dominant mutation. So capital H, small h, that is one parent which has got the disease, and small h where the other parent is not having the disease. So capital H, small h and small h. So capital H, small h, small h. So 50% of the offsprings will have the disease. Now suppose if the genotype is capital H, that is also a case of dominant mutation crossed with the parent which is not having the disease. That is small h, you will get all the progeny as having the Huntington's disease. But the option is being given for 50% only, not as 100%. So the answer should be, so the one of the parents should be heterozygous, only then you will get 50%. So answer is option B. Now next one. Phenylketonuria, it is caused by a homozygous recessive mutation that prevents the metabolism of phenylalanine. A man with sister who has phenylketonuria marries a woman with no family history of ketonuria. What is the probability a man with a sister who has phenylketonuria? So sister is having phenylketonuria but not the man. He marries a woman with no family history of ketonuria. What is the probability of them having a child with ketonuria? Assume no new mutations occurred in the parents. So now the man is not having a phenylketonuria, but his sister is having phenylketonuria. So he can have two genotypes, that is either capital P or capital P small p. Okay. So now what is the probability of them having the child? This man marries a woman who has no family history of phenylketonuria itself. So he will be capital P and woman will be capital P. So they will get all offspring as capital P because it is a homozygous recessive mutation. Okay, wait. Before coming to that, all the offsprings will be of capital P, that is without phenylketonuria. So 0%. Now his genotype, if it is capital P and small p, because he has got a sister who is having phenylketonuria, so chances of him getting a heterozygous genotype is common. So capital P small p into P, capital P, that is the women who has no family history of ketonuria. These are the gametes produced and these are the offspring. Even if that is the condition, you are not going to get a child with phenylketonuria because it is a homozygous recessive mutation. It has to be present as small p for having the disease. So the answer is option A, that is 0%. The next one. Photoreceptor cells present in eye that become active in bright light and play a role in the daytime vision are corn cells. Answer is option B. G protein coupled receptor is a monomeric protein. Answer is option D. How many GTPs are hydrolyzed during the initiation of eukaryotic protein synthesis? Only one. Answer is option A. And match the following with their respective effects. Carcinogen is neoplastic transformation of eukaryotic cell. Clastogen, it is causes fragmentation of chromosome. And teratogen, it results in developmental abnormalities. And mutagen, it causes mutation. So answer is option B, that is A3, B4, C2, D1. Then given below are different type of nucleic acids. Select the option which represent the nucleic acid that can be precipitated by trichloroacetic acid. So first and foremost, TCA, that is trichloroacetic acid, it will precipitate nucleic acids longer than approximately 20 nucleotides. So double-stranded DNA of 200 base pair, that will be precipitated, and RNA of 200 nucleotides, that also will be precipitated. So A and B, that is option A, will be correct. Whereas single-stranded DNA of 15 nucleotides and RNA of 15 nucleotides will not be precipitated because nucleic acids longer than approximately 20 nucleotides will be precipitated by trichloroacetic acid. Then arrange the following proteins in the order of their appearance during homologous recombination in E. coli. Rec A, Rec B, C, D, SSB, RUV, AB, RUV, So the arrangement is Rec B, C, D, SSB, Rec A, RUV, AB, RUV, Then most prokaryotic mRNAs. Most prokaryotic mRNAs have multiple Schindel-Garneau sequences and have 5' methylated guanosine, no. Transcription is uncoupled, no, it is prokaryotic, so it is coupled to translation. Then produce multiple proteins, that is also correct. So 1 and 4 are only correct, so the answer is option D. Then given below are some covalent modifications of proteins. Select the option that represent the covalent modification that impede protein mobility in an SDS polyacrylamide gel electrophoresis. So summation will impede, then ubiquitination also, as well as phosphorylation, but not racemization. So answer here is 1, 2 and 4 only. Then glucose breakdown occurs in actively metabolizing muscle tissue. Some of the enzymes involved in the process are listed in column A. Select the option that matches the enzymes with their features. So A, that is phosphofructokinase, it is irreversible reaction. Glyceraldehyde 3-phosphate dehydrogenase, it is by contains a free SH group at active site. Then pyruvate kinase reaction, substrate level phosphorylation, and lactate dehydrogenase is an iso enzyme. So answer is A3, B4, C1, D2. Answer is option A. Given below are some enzymes. Which of the following enzymes are required for bacterial DNA synthesis? So bacterial DNA polymerase 3 is required, ligase is required, telomerase is not required, whereas pyrophosphatase is required. So answer is 1, 2 and 4 only. Answer is option D. The next one. A scientist is working on repressors to regulate gene expression in a bacterial cell. Which of the following statements are true about repressors? Repressors are regulatory proteins that block transcription of mRNA, as you can see in the case of lac mRNA. Then repressors bind to a portion of DNA called the operator that lie upstream of the promoter. I think it is lies downstream of the promoter. Okay. Then the binding of regulatory proteins to the operator prevents RNA polymerase from the binding of the regulatory protein to the operator prevents RNA polymerase from binding to the promoter, thereby inhibiting gene expression. That is also true with regard to repressor. So 1 and 3 are correct only, are the only statements that are true. So answer is option C. The next one. The electronic configuration of iron in deoxyhemoglobin is dash and hence dash. So the oxidation state of iron in deoxyhemoglobin is Fe2+. So iron without any oxidation state, it is having this particular electronic configuration, that is 4s2 3d6. But Fe2+ state, it is not having the 4s orbital. So 2 electrons are lost. So 4s2 is not there. It is 4s0. So it is answer is 70. Answer is option B, and it is 3d6, 4s0 3d6, and it is paramagnetic. So answer is option B. The next one. The enzyme kinetics equation Kcat by Km is equal to V by concentration of S into concentration of E holds true for which of the following condition? So it is when Km is very much greater than the substrate concentration. Answer is B. Then what is the difference in the classical pathway of antigen presentation if plate A of macrophages are infected with live viruses while plate B of macrophages are treated with heat-killed viruses? So the answer here is plate A macrophages will present viral antigens via both class 1 and class 2 MHC molecule, because plate A macrophages are professional, not plate A, but macrophages are professional antigen presenting cells, and they will express both class 1 and class 2 MHC molecules. So the answer is option A. Then given below are statements related to transport of ions. So transport of sodium ions and glucose across luminal membrane of the epithelial cell in intestine, pumping of K positive inside the cell by Na plus K minus ATPase, pumping of Na plus inside the cell by a Na plus Ca plus transporter, and acidification of intracellular compartment by a H plus ATPase. Which of the phenomena are primarily due to secondary active transport system? Now, secondary active transport is not powered directly by the energy released in the cell metabolism, and there are two kinds of secondary active transport, that is counter transport and co-transport. Now, counter transport in which two substrates cross the membrane in opposite direction, and a well-studied system is that which pumps one calcium ion out of the cell in exchange for 2 or 3 sodium ions in. So transport of sodium, wait. So statement 3, that is pumping of Na plus inside of the cell by Na plus Ca plus transporter. That is one example of secondary active transport. Then there is another type of secondary active transport, that is co-transport, in which they cross in the same direction. So during the absorption of nutrients, for example sugars and amino acids are removed from the intestine by co-transport with sodium ions. So option statement 1, if you see, that is transport of sodium ions and glucose across luminal membranes of the epithelial cells of intestine. That is also a secondary active transport. So the answer here is option A, that is only 1 and 3. Then which of the following amino acids can be converted to both pyruvate and acetoacetate? So answer is option B, that is isoleucine and phenylalanine, as well as isoleucine and tryptophan. Answer is option. Then next one. Identify the correct statement of action potential of nerve cell. That is option Na positive ion in causes depolarization and K positive ions out causes repolarization. Answer is option C. Then next one. The following statements are linked to microtubule functions such as dynamic instability and catastrophe. So microtubule exhibit dynamic instability during which tubulin subunits will both associate and dissociate from the plus end of the protofilament. So that is correct. That statement is correct. So 1 is correct. Now, a microtubule catastrophe event manifests itself by change in the length of the microtubule and establishes equilibrium as shown in the plot below, in which A is catastrophe, B is rescue and C is equilibrium. But actually when you draw the length of microtubule to the time, you will get a graph like this. But A and B are catastrophe and C, and if you see over here this region, it is rescue. So statement 2 is not correct. So the answer is option A, that is only 1 is correct. Now this one, this question I don't know whether I am right or wrong in my answering, but anyway this is the way you have to do it. I didn't get any of the answers from the options given. I got my own answer, and after cross checking also I found that my method was correct, but I am not sure about it. So if you have anything with any comments to be given for this particular question, you can happily give it in the comment box. Then given that Plasmodium genome is 80% AT rich, what would be the average length of BAM H1 digested genomic DNA fragments? Now it is 80% AT rich, means it is 20% GC, not rich. Okay, I made a mistake over there. So 20% GC. So AT will be 40% each and GC will be 10% each, or you can say it is 0.4, 0.4 and 0.1, 0.1. Now the case of BAM H1, GGATCC is given. So probability of finding G at position 1. So at this position, if you want to find G, what is the probability? It is 0.1. Now if you want to have probability of finding G at position 2 also, then also it is 0.1. And if it is A, you have to find, then it is 0.4. For T also it is 0.4, and for 5 and 6 it is 0.1 each. So the probability of finding the BAM H1 sequence in the DNA is multiply all these probabilities and you will get 0.0016. Then average length of fragment will be inverse of this, 0.0016, that is 1 by 0.0016, that is 62 kilo base pairs. So that option is not given in your options for the exam. So you can give your comments in the comment box. Then next one. Choose the option that correctly matches the information from the given column. So topoisomerase 3, it is ATP dependence is no, and it is single stranded, wait, and single stranded DNA is plus or minus, change in the linking number is plus or minus 1. So the answer is option B. Then a mutant bacterial gene contains a stop codon in between its coding sequence. Yet it makes normal protein. This is most likely due to suppressor tRNA. So a suppressor tRNA is a tRNA with a mutation in the anticodon that allows it to recognize a stop codon and insert an amino acid in its place. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.